Hello, everyone, and welcome to this session of ACS Science Talks, Connecting the World Through Science. This is the virtual lecture series, Scientific Talks by Specialists on Specialized Topics for a Specialized Audience. I'm Neha, and I'm pleased to be your host for today's broadcast of ACS Science Talks. I'm joined by my colleague, Dr. Sharmishta Das and Dr. Reshmi Thomas, who will be moderating today's live Q&A. Some announcements. This session will be recorded live and would be placed in the ACS Science Talks library. In case you face any technical difficulty, you can reach out to us in the chat box. ACS Science Talks are an interactive program, and we would love to get you involved in the discussion. You can share your thoughts and questions in the Q&A panel. The moderator will take up these questions with the expert during the Q&A session. You can use the chat to introduce yourselves and say hi to our expert. Before we begin the session, a brief message from the American Chemical Society, ACS. In its efforts, ACS provides a variety of resources. One of the flagship ACS resource is the ACS meetings. Just like ACS Science Talks is a platform by ACS to connect researchers, ACS meetings aims at unifying the scientific community. Every year, active researchers and professionals from across the globe come together to share ideas and advance the scientific and technical knowledge. ACS meetings are regularly attended by thousands of science professionals every year. As we mentioned about the goals and mission of ACS to support the global scientific enterprise, ACS has taken the challenge of the COVID-19 pandemic and converted it to an opportunity. ACS meetings have embraced the hybrid model, which is helping us to enhance the networking opportunities by combining in-person interactions with virtual networking platforms, by promoting inclusion and accessibility by removing geographical barriers. It also enhances representation by reaching out to a diverse range of presenters and attendees. It provides flexibility and convenience by accommodating different preferences and circumstances. It increases engagement and interaction by fostering a dynamic and inclusive conference environment. It also provides continued knowledge by sharing opportunities, ensuring continuity of knowledge sharing during unforeseen circumstances. And lastly, it promotes sustainability and cost effectiveness by eliminating the need for travel and accommodation expenses and associated carbon footprint. Today, we are celebrating our 150th ACS Science Talks, and I would like to share the journey for the same. Thank you so much. It is because of you we have come this far. Moving ahead, our speaker for today is Professor Debashree Ghosh. Professor Ghosh obtained her MS degree in Chemical Sciences from the Indian IISc in 2005 and PhD degree from Cornell University, USA in 2010. She joined CSIR NCL as a senior scientist in 2012, subsequently moving to the Indian Association for the Cultivation of Science. Currently, she is a professor at IACS. Her research focuses on the development of polarizable force fields and their hybrid frameworks with excited state electronic structure methods geared towards understanding the photophysics of biological systems. She has been recognized by several awards, including the Annual Medal of the International Academy of Quantum Molecular Science in 2021 Serb Power Fellowship in 2022, and Walter Kuhn Prize awarded by International Center for Theoretical Physics in 2022. Thank you, Professor Ghosh, for joining us. The stage is all yours now.
First, let me start by thanking ACS for giving me this opportunity. And in fact, I have listened to these science talks for quite a while, and therefore I'm even more excited to be a part of it. I'm honored and excited. So today I'm going to talk about the photophysics of the skin pigment that is present called melanin. But before we go into photophysics, let me start from something which is much more accessible to us, which is chemical reactions. We are all chemists here. And whenever we think about chemical reactions, it is a picture such as this potential energy diagram that comes to our mind. And along the reaction coordinate, essentially, if you have an exothermic reaction or an endothermic reaction, you will have some kind of a delta G, a positive or a negative value, and some barrier that it needs to cross in order to perform that reaction. That barrier, in many cases, is quite high. Let's say this reaction is something like a cis-trans isomerization, but it's a very simple reaction. We have always looked at things like that. And we also know that most cis-trans isomerizations are fairly high in energy barrier, as in you have to pay a large cost to surmount that barrier and come away from the potential energy well of the reactant to the potential energy well of the product. So you have to climb that potential energy barrier. However, if the same reaction, again, a cis-trans isomerization in this case, here we have a stilbene molecule, and we are looking at the cis-trans isomerization of stilbene. If you look at the barrier in the ground state, as I mentioned before, it's going to be fairly high. It's about 60 kcal per mole. So if you are going to be performing that reaction on a lab benchtop, you will possibly need a lot of energy, lot of heat, or some other catalyst or this or that. However, what one can do is excite that molecule with light. Once the molecule, in this case, the cis-stilbene, if that gets excited by light and goes to its first excited state, then thereon after, the potential energy surface is reasonably downhill. There are no such big barriers, as you can see, and it kind of rolls downhill and reaches a position where these two, the two potential energy surfaces are reasonably close to each other. Sometimes whenever they're close to each other, there can be non-radiative pathways through which it can come down to the other potential energy surface. In this case, from the excited state to the ground state potential energy surface. From there on forward, now you are on top of you are sitting on top of the transition state. And this you have enabled through this light excitation by light. From this transition state, of course, there's going to be some percentage of some probability of it going back to the cis state and some probability of it going to the trans state. Since the difference in energy between the cis and the trans in this case is not very high, we would expect that from this transition state, the probability of going left or right is almost equal. So that's why we have written over here a kind of 50% probability. And so you basically can see that the cis-trans isomerization can be done with much less harsh conditions, no high heat required, no fancy catalyst required, and so on. So in some way, excited states open up pathways that would otherwise be insurmountable via ground state reaction mechanisms. That's essentially what my group is involved in looking at. Not just in stilbene, but many different molecules show these kind of excited state reactions. And again, as you can see, the difference in the ground state reactivity and the excited state reactivity is quite spectacular. Therefore, the chemical intuition or the chemical knowledge that we have for the ground state processes can be quite different from those that we need for the excited state processes. So it's in these kind of questions we are trying to give some plausible answers towards. So in the previous slide, I had actually shown a small reaction and a kind of a clean mechanism where everything is known, or at least much of it is known. However, if you look at the potential generic potential energy surface of any ground state and excited state of a molecule, this is quite complicated, and there are very many different situations that one should consider ideally. So let's look at this reaction potential energy surface. So here again, we are starting from the reactant. The reactant is happily sitting in the ground state minima. From there, if you shine light on that, it can go to the optically bright state. In this case, we have written it as the S2 state. It could be the S3, it could be the S1, it could be whichever state is optically bright. Once it reaches the optically bright state, the logical thing which the molecule will start doing is follow the gradient of that potential energy surface. And along that gradient of the potential energy surface, it can encounter very many different kinds of reactions, either radiative or non-radiative. Non-radiative processes such as crossing between two same spin excited states, for example, the S2 and the S1 state, these are referred to as conical intersections. The molecule can encounter these kind of situations of the potential energy surface. It could also encounter crossings between the singlet state and the triplet state, because notice again, beneath any singlet state, there's going to be, in most cases, quite a few triplet states. And so the molecule can encounter those singlet triplet crossing points. And these crossing points can again become, ideally, we kind of think that there's no singlet triplet crossing, but singlet triplet crossing or intersystem crossing does happen when there are high spin orbit coupling situations. So those processes are going to happen in certain select cases. Furthermore, apart from these non-radiative processes, there's going to be radiative phenomena such as fluorescence, phosphorescence, and so on. Finally, another thing we should keep in mind is while this molecule is kind of rolling down and sometimes rolling up a bit also in these potential energy surfaces, it will encounter crossings, and in those crossings it can encounter situations where instead of leading it back to the original ground state minima where it started from, it can lead it to some other minima. That is, for example, in the last slide, as we saw, the cis-trans isomerization. We started from the cis state, excited it, it came down to some kind of a crossing or crossing-like situation, and then it rolled down 50% to the original product, original reactant, but 50% to the product, which was the trans isomer. So these kind of products can also form along the way. The study of this field is actually a rapidly evolving one. And this, as you can imagine, would relate spectroscopy for sure, but it also involves a lot of electronic structure calculations, dynamical calculations such as molecular dynamics. And in this case, the molecular dynamics will not be only for the ground state, but also for the excited states that are involved. So that's a fairly complicated looking scenario that would happen in its entirety. So how does one start by looking at it? And here I should have mentioned, I am a theoretical chemist and a computational chemist. And so my approach or the directions I'm going to show are mainly going to be based on the computational approaches that are involved. So the way to solve this kind of insurmountable looking problem is essentially based on electronic structure of these different excited states. And so whenever we are talking about potential energies, that means I'm looking at different geometries of the molecule. And at those different geometries, I am looking at the electronic structure of the different excited states or the ground state that is involved. So I would obviously have to start from a Born-Oppenheimer approximation, which basically says that if you have nuclei and electrons all in this situation, which you of course have if you have a molecule, the nucleus is moving much, much slower than the electrons. And therefore, we are kind of freeze framing it at different nuclear situations and calculating its electronic structure. So that's what it allows us, Born-Oppenheimer approximation. Once we have this approximation set up, and from that approximation, we essentially have the electronic Schrodinger equation. The first approximation, next approximation one takes is the mean field approximation, which is essentially our molecular orbital theory. We use Hartree-Fock theory to solve for the molecular orbitals. And in this situation, we do not have any correlation other than the same spin correlation that is involved. Other than that, we don't have any other correlation here. Since we don't have this correlation, but electrons, of course, are correlated because electrons are charged particles. They will try to avoid each other instantaneously. So in order to account for the correlation, which we disregarded to begin with, we would try to correct for it, ideally using some kind of a perturbative correction, such as MP2, CCSD, etc. So this is essentially going to be our main workhorse in any of the calculations, any of the analysis that I'm going to be talking about today. This is where things get rather more complicated. And I will basically start with one of the failures of the approximations that we talked about in the last slide, as in things are not as easy as they sound. And the reason for that, as I'm going to show, is the breakdown of the perturbative correction in the last point of the last slide. So if you take a water molecule and pull its bonds apart, so that's essentially you are moving away from the nice minima of the potential energy surface to some other place where it's it's not a minima, it's some other part of the potential energy surface. In that situation, so the exact calculation is shown over here with the black, marked in black. But if you do some kind of a perturbative approximation, the names of which I mentioned in the last slide, MP2, or let's say not perturbative, but CCSD-like approximation, you will see that there's something really funny going on. Because you see, if you are pulling apart a bond, what do you expect? At the end, when you have, in the asymptotic limit where you have only the radicals that are formed, you would think that the potential energy will smoothly go to non-interacting limit, where none of the radicals are interacting with each other. However, for the approximate methods, you see such sudden bumps and then kind of dip into values which are lower than the exact value. In other words, if I look at the errors that are encountered in such approximate calculations, you see that the errors are going to, well, probably negative infinity, where I have broken the bond and I have formed radicals. That's a really disastrous situation for a chemist, because ideally we would like to break bonds and make bonds. And so if the theory only works near the minima of the potential energy surface and not at other situations, that's a difficult problem for us. Furthermore, these effects show up even more in case of excited states, radical species, polyradical species, transition metal complexes, polyenes, graphenes, with large conjugations, and metal clusters, and so on. So essentially, this kind of shows up in all the, I would say, exciting chemical problems, anything that you can think of. And the reason for that, actually, that is also quite known. It's essentially in the minima, when you are at the minima of the potential energy surface, you have a nice separation between the highest occupied molecular orbital and the lowest unoccupied molecular orbital. So the frontier orbitals are reasonably separated because you have a bond, so the HOMO will be much more stable than the LUMO. And you essentially are in the regime where our mean field approximation or the molecular orbital single reference molecular orbital theories work spectacularly. However, when you are pulling the bonds apart, or you are going ahead in some other parts of the potential energy surface, the bonds are getting weakened. So the energy stabilization of the HOMO and the destabilization of the LUMO is getting smaller and smaller. And you have situations where you cannot say that a single electronic configuration is the predominant one. In these situations, there are very many different electronic configurations that one would need to consider to have quantitatively accurate numbers. So that's what's going on. And therefore, one needs to include multiple electronic configurations and therefore multi-reference or multi-configurational methods, at least in the valence space of the molecule. This is a difficult proposition. I'm not going to belabor this point because today I'm not going to talk about the methods, but rather the results we obtained from some of the methods. There have been some traditional multi-reference methods such as active space-based methods. Again, there's no reason to complicate it. It's as if you basically consider the frontier orbitals as active orbitals, you can circumvent quite a bit of these artifacts that you see in these mean field approximations. So that's what we will use or we have used for the rest of the results that I'll show. There are some methods that are developed in our group because as the number of active orbitals increase or the valence space increases, the number of references that one needs to consider increases. And in those cases, there are other methods or newer methods that require to be developed. Our group is actively involved in these method developments. Those are the things I'm not going to talk today. In any case, using the methods that we have at hand, we have actually looked at, even before I go into the problem of melanin, let me kind of give a glimpse of what else we have been spending our time on. We have looked at singlet fission processes, which is essentially where you have a material system where there are multiple chromophores that are in close vicinity of each other. And when light shines on these molecules, one or the other of the chromophore can get excited. And since the molecules are in such close proximity, they can share their excitation and form two triplets from this one singlet state. And since these two triplets are coupled triplets, they do not violate the spin selection rule. So this is indeed a very fast process. And in this, we have been looking at the mechanisms and electronic structure of the states that are involved in singlet fission. We have also been looking at some of the excited state processes in polyaromatic hydrocarbons. There are many polyaromatic hydrocarbons that people have synthesized along the way. Different motifs, different structures of conjugation and so on. And we have tried to look at how different aspects of these elongated or long-range conjugation changes the singlet triplet gaps or ground state to the first excited state gap and things like that. So that's another aspect we have looked at in our group. But let's come back to what is the main topic of today's discussion, which is melanin. So melanin is a strange molecule. This is the spectra of melanin. And as you can see, it's extremely uncharacteristic of any other organic molecule. It's completely flat, completely featureless, and monotonic. That is great for human beings because what it means is that it can absorb all wavelengths of UV and visible along the way. It essentially soaks up any sunlight that is incident on a human body or a plant or animal or whichever is using these melanin molecules or melanin species to protect itself from sunlight. So that's fantastic. However, what makes it difficult is we generally tend to use these spectroscopic features to identify the chemical composition, the chemical bonds, how that molecule looks, the chemical structure of that molecule. And because of this strange looking spectra, the melanin structure has continued to remain elusive and a mystery to people. There has been quite a few models that has been proposed by different groups. As you can see, they're significantly different from each other. However, what all of these groups have agreed on is that once these oligomers of melanin are formed, they kind of form stacks on top of each other in a non-covalent way. And that's essentially the full structure of melanin as we know it. But the oligomeric structures remain still extremely elusive. What is known, however, is what are the monomers of melanin, especially eumelanin. Eumelanin is the brownish black pigment. There are other kinds of melanin, such as pheomelanin, neuromelanin, and so on. I'm not going to talk about those. I'll only talk about eumelanin, which is this brownish black pigment. And it's actually especially good at absorbing sunlight and still protecting the body from all the harmful effects of sun. And the melanin, the eumelanin, is formed of dihydroxyindole. You can see this is an indole moiety with two hydroxyl groups. There are also some substituted form of this dihydroxyindole with substitution of carboxylic acid or even actually other groups in this position. Along with these basic units, there are the oxidized forms of it, such as the monoketo form. Here you see this is a monoketo form, or the diketo form. So there's quite a bit of diversity present in the monomeric structures of melanin. That's essentially why its structure is even more difficult to figure out. So the questions that we are going to ask, or rather answer today, are what are the photoprotective pathways in melanin and how does melanin get its color? So as a quantum chemist or a computational chemist will do, we will start with the monomeric unit. And this work was started by a former PhD student in my group, Debashree Ghosh. She did phenomenal work in figuring out the different pathways of photoprotection in melanin. So in dihydroxyindole, if we look at the Frank-Condon region, or the region where the ground near the ground state minima, there are two excited states that are close to each other. The first state, the S1 state, is the optically active pi star state or optically bright pi star state, and the second one is the optically dark pi sigma star state. Now, when we started looking at these molecules, there were some earlier papers that suggested that some of the important degrees of freedom in this case could be the NH bond elongation and OH bond elongation. So we tried to look for conical intersections or non-radiative pathways along those degrees of freedom. And if you elongate the NH bond, what you would expect is this sigma star orbital to have a large change in energy. The pi and the pi star would be relatively insensitive to this change. And that's exactly what happens. So we encounter a conical intersection that has pi star that between the pi sigma star state, and the pi star kind of remains as is. So the total potential energy surface kind of looks like this. However, when I look at the specific quantitative features of this, I see that this conical intersection is a reasonably high barrier one. And also it is high energy itself, as in the conical intersection itself is higher in energy. Since these excited state processes are always really fast because there's fluorescence which is always competing with it. If it's a slow process, it would fluoresce and come down the molecule. So it has to be faster than fluorescence. So we didn't think that this pathway would really be of any consequence. So we went ahead and looked along the OH bond elongation, and there we see that we have a similar conical intersection. This time, however, thankfully, it is a low barrier and a low energy conical intersection. So energetically, this conical intersection would be easier to access. And then what we did is we looked at the topology of the potential energy surface near the conical intersection. So essentially the directions where the energies, the degeneracy of the energies are lifted, those directions and the conical intersection around that. And what we noticed is that the NH elongation, which, as I mentioned, had a large barrier and would probably never be accessible, turned out to be the one that led to photoprotection. And the OH elongation, which had a smaller barrier, so ideally, energetically, it would be easier to access, it led to both photoprotection and photoproduct formation. Now, this was confusing. Of course, melanin does not form some other product and degrade that quickly. Because we have melanin in our skin, it protects us throughout our lifetime in most cases without undergoing any photo damage whatsoever. So definitely we were missing out some other directions of quenching or the energy, which we haven't yet looked at. So we went ahead and what we did is perform some dynamical calculations. So previously what I was showing were mostly static calculations. Here we went ahead and did dynamical calculations. And by dynamical calculations, what we did is essentially surface hopping calculations, really simple surface hopping calculations with the already obtained potential energies with any active space-based methods. With that, we saw that as expected, that NH elongation mode never showed up. There were a few trajectories which showed OH elongation, which is what we expected. And there were many trajectories which showed that the whole molecule puckered into this strange form. So in fact, deactivation in most cases happens through a non-planar pathway, not this planar single bond elongation kind of a reaction. So we then, after the dynamical studies, we went ahead and again did the potential energy surface along that ring puckering mode. And along the ring puckering mode, now I would expect that the pi and the pi star would have maximum change. The sigma star will not care what would happen, what ring puckering is going on. That's exactly what happens. So now I see a conical intersection between the pi star excited state and the ground state, and the sigma star, pi sigma star states just remains as is. Here is the potential energy diagram along that mode. And lo and behold, this one does not have any barrier. That's why we saw so many of those in the trajectories. And if I looked at the topology around the conical intersection, this showed only photoprotection modes, no photoproduct formation. So that's essentially, we are slowly uncovering the different modes that melanin has to save itself from sunlight. So kind of to re-summarize or reiterate what we have understood till now is for this dihydroxyindole moiety, we have two modes. The first one is planar, which has a small barrier, but leads to both photoprotection and photoproduct. And the other one is non-planar. It has no barrier, so it's really fast. Most of the reactions will go along that way, and it leads to only photoprotection. So this minor channel and major channel, the minor channel, as I mentioned, is photoprotection and photoproduct. And this is another thing we noticed over here. It is not just the photo product or that is the some other thing, something that is going to degrade melanin. In fact, the photo products are hydrogen abstracted oxidized products, which are, remember those MKI or the monoketoindole and the diketoindoles that I showed before. So in fact, what is happening is melanin in one form is getting interconverted between the different forms that are present. So the photo products are itself are also other forms of melanin, which is fantastic. And as I mentioned, the major channel just completely deactivates into the original DHI. We have also done studies on the monoketo and the diketo form, which I'm not going to go into details, but I'll just tell the full picture once and for all. So from the minor channel of the dihydroxyindole, we have the diketo form and the monoketo form. In fact, there are two kinds of monoketo forms that are possible, right? They're different. If the one of the hydroxyl group gets oxidized versus the other one, you have two different forms. And on shining light on the diketo form, we notice that it again comes back completely to itself. So it is photoprotective of itself. And the monoketo forms actually have a pathway of interconverting between each other. So you see over here, there are very many things going on, but each of them is a closed loop. There is nothing that is forming a degraded product. The product itself is actually one or the other form of melanin. That's essentially how we believe melanin protects itself from harmful UV radiations of the sunlight. But at this point, we also looked at effect of some small molecules on the photophysics. And for that, we looked at water, since water is one of the most important biological molecules, small molecules. And here what you will see is quite spectacular. So in fact, there's not one sigma star state that is orbital that is close to each other, close to the pi star state orbital. There are quite a few sigma star states. And these sigma star states have different relative energy depending on where the water gets hydrogen bonded. So you see these different states are actually rearranging themselves among each other depending on where the water is hydrogen bonded. And depending on that, it's those different modes, whichever is closest to the pi star state, that is becoming the secondary most important direction of photophysics. So that's one thing we have studied. We have also looked at the effect of dimerization. The reason is because, see, I already mentioned melanin does not stay in its monomeric form. It's in some kind of an oligomeric form. And so we basically wanted to build those blocks one after the other and see how, or if the photophysics changes considerably or not. And in fact, the effect of dimerization, we saw there was another mode that was opening up, which is essentially the excited state proton transfer mode. So, this proton transfers from here to there, and that becomes one of the other major modes of deactivation or non-radiative deactivation in dimers. And we have actually even looked at higher oligomers, and we see that either it is these monomeric modes or these dimeric modes that comes about in higher oligomers. So there's no new story after dimer. Dimer is kind of the end of cool photophysics going on. So that's essentially what we have found from bottom-up studies of the melanin monomers and dimers and their photophysics. But in the beginning of the talk, I kind of mentioned that there are two questions we are going to ask and answer. The first one is, of course, what I answered before, that is, what are the photoprotective pathways in melanin? you saw that the photoprotective pathways are interconversion between each other, and also excited state proton transfer, which opens up only in dimer or other higher oligomers. But the second question, and one that has confounded people for a long time, is how does melanin get its color? So actually, this was a work by my senior PhD student right now. He basically kind of combined computational chemistry and machine learning and answered this question to a large extent. I'm not, again, as I mentioned from the beginning, I will not go into the details of the method so much, but I'll just give you a sense of what was being done. So to reiterate the question, normal biological chromophores would have a spectra like this right-hand side one. There would be certain features, shoulders, humps, this and that, and maxima. And depending on the position of the maxima, you basically figure out what is the chromophore that is present. But in melanin, there is nothing like that. This is the red one is the eumelanin spectra. The blue one, the pheomelanin spectra. Both of them are relatively featureless. And so the question now is to answer, basically, if I have a narrow range of the wavelength, let's say 500 to 510 nanometer, then what is the chromophore that is excited there? Or what is the chromophore that is leading to excitation absorption in that region? So those kind of questions. And this is a problem that I am just showing some examples of how difficult it has been to solve this. This is in fact an inverse design or an inverse learning kind of a problem. So what we have at hand is the spectra, and what I want is from that spectra to figure out what is the chromophore that is responsible for absorbing at that wavelength. These are, after this combined inverse learning technique that Orpon has discovered, what we found is if we train our question along certain small ranges of the wavelength, these are only for example purpose. If we say 280 to 290, there are these different structures. And what's most important is these structures are spectacularly different. So there's quite a few different chromophores that absorb in different narrow wavelength ranges of melanin. So that's essentially the point I would like to make. So essentially, melanin has large amount of heterogeneity, and this structural heterogeneity that is present in melanin has two roles. One, of course, to absorb this UV light across this large range of wavelengths, and then to take it and interconvert the products among themselves to protect the melanin structure. This was published in this Advanced Science News article. You can take a look at that. And of course, every time I show this, I'm always very pleased to see that computational chemistry happens to be the key in this case. The reason for that is it's actually melanin is a very difficult experimental, different molecule to study experimentally. That's why it's a computational chemist playground. So to conclude, we have predicted the exact structure of melanin, at least certain kinds of melanin in certain ranges of the wavelengths. And eumelanin spectra can be understood from a combined computational and machine learning technique. Furthermore, we have shown that interconversion of monomers is the key to photoprotection in melanin. We have studied various aspects of it. I needn't belabor that point anymore. But I can't stop before I acknowledge the main people who did the work. So the work I presented today was mainly done by Poulomi and Orpon. Both of them did fantastic. And this is my group kind of now, but I think there are other people who might even have joined as participants. Hi to you. And various collaborators from different, mainly spectroscopists, Jyotishman and Naresh. And Raghu helped us with the machine learning part, funding from various sources. And again, thanks to ACS for giving me this opportunity. Thank you.